Welcome to our Life Stage Research Review Group project. We chose to explore the controversy that surrounds whether or not exposure to cow's milk protein during infancy is a culprit in the onset of type 1 diabetes. We begin by reviewing some of the essential characteristics of what is known about type 1 diabetes. First and foremost, the exact cause actually remains unknown. The current theory is that it's an autoimmune disease, and this is based on an assumption that an infection or another trigger of some sort causes the body to mistake the beta cells of the pancreas for foreign entities, and thus they are destroyed. Obviously, the destruction of beta cells leads to pancreatic insufficiency. Because the pancreas is responsible for the production of insulin, which is necessary to sustain human life, it's clear that a lack of insulin production is going to lead to death. And therefore, this makes an understanding of the role of insulin in the body key to appreciating the devastating impact of this disease. Let's watch a brief video clip that helps illustrate.
Insulin helps your cells absorb the glucose in your blood, allowing it to be used as energy. A healthy pancreas releases a regular supply of insulin into your bloodstream. After you eat, your blood glucose levels rise, and your pancreas responds by releasing more insulin to move the glucose into your cells. Insulin acts as a key, opening up the cell so it can accept the glucose. In a person with type 1 diabetes, the pancreas produces little or no insulin. Without insulin, blood glucose levels rise. Without insulin, glucose cannot enter the cells and be used for energy. As a result, it remains in the bloodstream.
In order to understand what's to follow, we have to begin with a few basics. Current statistics estimate that approximately 3 million people in the United States have type 1 diabetes. Though most are diagnosed early in life, infancy through early childhood, diagnosis can occur at any age and does often occur in the 20s and 30s and even beyond. Typically, a diagnosis does not occur until insulin production has completely ceased. In other words, once the beta cells have been completely destroyed. So the evolution of that process takes time dependent upon the individual's disease progression, and once it fully manifests, the signs and the symptoms will become noticeable enough that one will seek attention and a diagnosis will be made. Genetic associations have been established, and they are one of the areas under focus for much of the current research. In order to appreciate those associations, you have to fully understand the genetic component. So let's make sure we're all clear that genes are essentially the blueprint for the production of proteins in the human body, and that those subsequently produced proteins function throughout the body in numerous capacities, all of which are clearly vital to human life. Unusual variations in the function of a gene results in altered protein building instructions, and therefore altered bodily functions. It is understood that gene variants do exist within individuals and are thus a factor in impaired bodily processes. What we're uncertain about is whether the variants are inherited or environmentally influenced. And when we talk in terms of environmentally influenced, we're referring to reactions to stimuli in our environment, such as viruses, dietary components such as nutrients or otherwise, or exposure to various toxins intentionally or otherwise. And so whether or not the variants that lead to this particular disease or other diseases under explanation are the result of inherited variants or environmentally influenced variations in the gene function is very difficult to ascertain, and that's why it's the focus of much study today. HLA proteins are at the crux of current exploration. The human leukocyte antigens, or the HLA system, is in essence a group of genes which code for the production of proteins involved with the function of the human immune system. The resulting proteins serve in various disease defense roles, including that of antigens and leukocytes. HLA proteins generate antigens and leukocytes to specifically help the immune system to recognize a cell as either part of the human body or as a foreign entity, and as a result, foreign entities are destroyed. Thus, gene variants in the HLA region of DNA may be responsible for genes generating immune system responders which mistake pancreatic beta cells for foreign entities, and therefore this could be, in essence, the path which leads to the onset of type 1 diabetes. So this leads one to wonder, what sorts of environmental stimuli might be the catalyst for the gene variant that sets this pattern of events in motion? Current research is exploring the relationship between exposure to cow's milk proteins during infancy and the etiology of type 1 diabetes. This is a controversial area of study with mixed findings, and that's what we're going to show you today. This particular quote from a recent publication sums it up quite well. Epidemiological studies suggest that exposure to cow's milk proteins during the first 2 to 4 months of age is associated with anywhere from a 1.5 to 2-fold risk of type 1 DM. This hypothesis is driving much of the research. So, a commonly posited hypothesis is that bovine insulin in cow's milk introduces primary immunization to insulin in infants who are exposed to cow's milk formulas at a time when gut maturation is clearly incomplete, and thus subsequent autoimmune activation against insulin-producing beta cells ensues. Let's take a look at some of the research. The first publication that we reviewed is a study which evaluates the early development of antibody-mediated immune response to cow's milk protein during infancy and the relationship to the development of type 1 diabetes in childhood. The methods involved infant subjects in Finland, and these subjects were those with an increased risk of type 1 diabetes due to HLA and at least one first-degree relative with type 1 diabetes. The study employed the use of 94 infants. It was a randomized study, and they were to receive supplement to breast milk, traditional whey protein formula, or hydrolyzed casein formula until roughly 6 to 8 months of age. They were split into 3 subgroups: those diagnosed with type 1 by age 7, those having at least one disease-associated autoantibody, and those who were autoantibody negative. Blood samples were evaluated and analyzed, and the findings are what you see here. We'll notice on the left-hand side of the screen is an outline of the various characteristics. You then have each respective column. Those diagnosed with type 1, those with at least one associated characteristic, and then you have those that were in the control group, and you see the percents respectively. Some of the limitations worth noting are that of the small sample size and the fact that the study does not specify the amount of breastfeeding versus the control and test formulas. Formula was simply given when breast milk was not available. The results, what they found is that children diagnosed with type 1 diabetes and who had a minimum of one disease-associated antibody had a higher IgA antibody binding presence as compared to the autoantibody negative children. Those later diagnosed with type 1 had higher IgG antibodies from 3 to 18 months and higher IgA at 9 months. The implications here are that this demonstrates that children who develop type 1 have increased immune reactions to cow's milk protein in early infancy, possibly due to small intestinal involvement with heightened gut permeability or delayed development of gut immunity. Ultimately, the implication is that delaying initiation of cow's milk protein may decrease the risk of type 1 diabetes in the minority children who develop the disease due to impaired dietary antigen immunity. The next study we looked at served the purpose of testing the significance of weaning infants to an extensively hydrolyzed formula to decrease the cumulative incidence of diabetes-associated antibodies. It was a randomized, double-blind study. It took place in multiple countries with a very large sample size. The participants that contained an HLA genotype and a first-degree relative diagnosed with type 1 diabetes were differentiated. The smell and taste of the formulas were designed such that they were kept consistent, so there would be no bias. And the mothers of the infants were encouraged to breastfeed. It should be noted that their diets were not modified in any way, however. The intervention lasted until the infant was 6 months old, and if the infant had not been weaned at this point, then they were weaned to their formula for at least 60 days from the 6-month point. Adherence to the intervention was modified by interviews with the family and analysis of cow milk antibodies in serum samples. So the measurements were from cord blood samples, and then there were follow-up samples obtained at 3, 6, 9, 12, 18, and 24 months, respectively. The primary outcome was the diagnosis of type 1 diabetes at age 10, and the secondary outcome is testing positive for 2 or more islet antibodies by age 6. As you look at the chart here, what you will see is that, again, it was a double-blind, randomized clinical trial with HLA-conferred disease susceptibility, so we're looking for variants in this region, and first-degree relatives with type 1 DM, so we're making the associations. You can see that the distribution of participants was roughly equivalent, half receiving hydrolyzed casein formula, half conventional cow's milk formula, and you see that the absolute risk of positivity for 2 or more islet autoantibodies was roughly equivalent. Limitations were that the results are not generalizable to children who do not carry the HLA genotype or who have a first-degree relative with the disease. And although the study has many strengths in a large sample, there are many previous studies that have been conducted similarly with contrasting results. The results for the study were different from previous studies done with a similar purpose. And so what we see here, again, is that they found that there was very little difference. So there was no clinically significant difference between the groups, and the implication could be to continue to promote breastfeeding for 6 months because of the strong evidence to support breastfeeding, and then to ensure the infant is receiving adequate nutrition through continued breastfeeding or weaning to a formula, because this study does not give enough evidence to state that either offers a protective factor against type 1 diabetes in high-risk infants. This particular study found that weaning to a highly hydrolyzed casein formula in infants with the HLA genotype and a first-degree relative with type 1 diabetes was not associated with a reduction in the signs of cumulative beta cell autoimmunity. The next study we looked at served the purpose of studying the association between early feeding events with the development of type 1 diabetes. The methods here included a very large cohort. This cohort was actually part of a completely separate study. That study was looking at the importance of supplementary feeding in hospitals on the later development of cow's milk allergies. All supplemental feedings in the hospitals were therefore known, and the babies were able to be randomized to receive one of 3 different formulas. So it was a huge asset to be able to use this cohort for dual purposes because there was so much monitoring in place and it was so accurate. So a portion of the infants were solely breastfed and a portion received supplementary formula before discharge from the hospital at the time of birth, and all were accounted for so that any differentiations could be taken into consideration. The mothers recorded the feedings of the infants prospectively, what they'd be doing at home, and at a later date, because the study took place in Finland and they have a mandatory nationwide diabetes registry, the researchers were able to access that registry a number of years down the road at the point when the children would have been 11 to 12 years of age and see how many of the children in that cohort had been registered. Therefore, they knew how many had developed type 1 diabetes. They identified 45 children from the total cohort that was in excess of 6,000 as having been diagnosed with type 1 diabetes by age 11 to 12, some point between infancy and that age. And they completed an analysis of that infancy feeding history data and compared the feeding history between those with and those without the diabetes diagnosis. You can see here the red asterisk represents the number of all children who contracted type 1, whereas the red pound sign represents the number of all children who did not. And so if you look on the furthest left side, the first 2 columns represent the percent based on cow's milk formula. You then have 2 columns representing hydrolyzed whey-based formula, 2 columns with banked pasteurized breast milk, and 2 columns representing the mother's own breast milk. So some limitations worth noting were that prospective studies of feeding practices may inherently be flawed due to changes that occurred in what the mothers had planned. Interesting though, this study really, again, lacked significant limitations because it was so unique in its access to detailed cohort data, such as exact knowledge of the early exposure to cow's milk formula, since this took place in the hospital and it was very clear as to what was administered exactly in what quantities, on what dates, and what types. So when we look at the results, we find that there was no association in the number of cases that developed type 1 diabetes when we're looking at the different groups who randomized either to formula or breastfeeding. In fact, the correlation between the consumption of formula in the hospitals and a reduced incidence of type 1 diagnosis by age 8 was a significant finding. However, by age 11 to 12, any such correlation disappeared regardless of what sort of feeding had taken place within those early days. Of the 45 infants who did develop type 1 diabetes, the proportion of those solely breastfed was equivalent to those who received supplemental formula during the same time period, 27% for both groups. Based on the introduction of foods, it was noticed that those receiving vegetables earlier in life experienced lower incidences of type 1 diabetes. Those who received cereal or grain products earlier showed no differences earlier or later, and those who received cow's milk formula earlier tended to display a lower incidence of type 1 than those who received it later. So, potential implications are that the optimal development of the gut-associated immune system and its regulatory functions may be stimulated by early exposure to food proteins, such as those in cow's milk, though this may well depend, once again, on the genotype of the given individual. So, in conclusion, these studies clearly leave room for further speculation. The first study found a positive association between cow's milk proteins and the development of type 1 diabetes. The second study found no clear association, one way or the other. And the third study found a positive association between the consumption of cow milk proteins and the prevention of type 1 diabetes. The limitations of the studies, however, further emphasize that there are many confounding factors that make it difficult to assess the correlation between infant feeding practices and the onset of type 1 diabetes. The realization that the cumulative findings infer is that the interaction of genes and immune responses to environmental antigens may well be driven by individual genotypes, and thus being able to forecast which genotype confers protection or susceptibility to any given condition is the mystery whose resolution is worth pursuing. We thank you for watching our presentation, and here are our references for your review.